Today I'm going to be talking about the application of genome engineering to facilitate point of lay sex sorting in the poultry industry. So I guess just as a disclaimer, I used to be in Mark's team down at CSIRO. So that's where I cut my teeth in poultry genome engineering. Before that, I did my PhD at UC Davis. Allison was there, where I was doing work in swine and in goats, so more mammalian genome engineering. But today I'm going to be talking about poultry, and currently I'm consulting for Hendrix Genetics. And so this is a project that's a collaborative project between Hendrix Genetics and CSIRO. Currently, current practice in the poultry laying industry is that, as people have touched on before, in certain sectors of agriculture, we only need one sex of the animal or the other. In the poultry laying industry, obviously, at the very end of the breeding system, you only need the females because only females lay eggs. You don't need all these males. And so currently what you have is a system where you have chickens that are hatched and you sex those males and females right at hatch, and you have about 7 billion males that are culled at point of lay. So that's a huge area of waste. Obviously, it's a huge sustainability issue, and it also presents a lot of ethical issues in the industry. And so it's something that the industry has wanted to tackle for a really long time, but obviously it's a really difficult issue to tackle because of a lot of technical feasibility issues in there. And so where do we stand right now? So we've got current industry practice, which is, there we go. So current industry practice is we take all these eggs, we put them in incubators, we hatch them, and then we sort them at the day of hatch into males and females. It's actually a very skilled process. People are paid a lot of money to be able to sort chickens. It's not very easy to do. Obviously that's, time intensive, labor intensive, and takes a lot of resources. In Europe, legislation has come along that has banned this practice. And so in certain countries, you can no longer cull male chicks at day of hatch. That's current practice, or that's currently enforced in Germany, and that's coming down the track in other countries as well. And so especially in Europe, there is a lot of focus on what are other technologies that we can implement. And so there are a variety of different technologies that are coming in at times like day 9 and day 14. So you're still incubating, you're taking all of your eggs, you're incubating them for a certain period of time. Day 9, you're almost halfway through embryo development. And so you can see here, there's a day 9 embryo. What you do is you take all the eggs out of the incubator, you're putting a needle in, you're taking a sample, and you're actually doing a hormonal analysis. And so that allows you to sort your males from your females. You sort those eggs, and then all the females go back in. But as you can imagine, this is millions and millions of eggs. You're going to have some attrition because you're sticking a needle in there. And so you're going to have losses associated with that, and just like the cost associated with pulling all these eggs out, doing all these hormonal analyses and putting them back in. And so while this is a solution that does fit the parameters that are needed in Europe right now, which is you cannot cull those male chicks, it's not necessarily a solution that we can roll out to the rest of the world because of the costs that are associated with it. And so the industry is really keen to come up with a solution that solves this problem, but doesn't take as much time and effort and labor and cost, because in Europe, the consumer might be more willing to accept the costs and the cost increases that are associated with implementing these technologies, but that's certainly not going to be the case in all jurisdictions. And so what we're looking to do is to develop technologies that allow you to sort the males from the females right at point of lay. So that means as soon as the eggs come out, you can say, this is a male, this is a female, have a non-invasive system. You don't have to put anything into the egg, leave the shell intact, and sort them all before you actually even put them in the incubator. And so you've got a lot of viability improvements here because, as I said, it's a non-invasive system. You're not sticking anything into the egg, but also you have a huge sustainability improvement because you're not incubating all of these eggs anymore. You're only incubating half those eggs. And so whether you're going to be able to increase production because now you have way more incubator space, or just lower your carbon footprint because you don't have to use as much incubation to get those females out at the end. So there's a lot of positives about being able to implement a point of lay sex sorting system. And so it's not a great picture, but or it's not showing up super well here, but this is basically what our proposed solution is. And so what we have is point of lay eggs that are carrying a fluorescent marker that allows us to sort them at point of lay. And so here you've got our gene engineered blastoderm just under regular light. You can just barely see that that's the blastoderm there. And so Simon did a great job of explaining how the blastoderm is sitting on top of that yolk underneath all that egg white. So that's, that's your developing embryo there. And then if we put it under the RFP filter, you can see that it's actually producing fluorescence. And so the idea is that we would be able to detect that fluorescence through the shell, allowing us to do the sorting. So what does this look like just as a schematic? So here, you've got all of these eggs here. They're coming through. You have no idea what they are, whether they're males or females, but inside you've got those blastoderms. And if you run them through a detection system, which, I say run them through a detection system as if that's easy, that's not easy. That's a really large technical hurdle that we are, currently addressing, but we do believe that the technology certainly exists out there to be able to overcome that. So, but yes, this is not a trivial part of it, but it is a very important part of it. Run them through a detection system that allows you to quickly, efficiently, and really cost effectively sort your males and sort your females. The current thinking is that, males, that just becomes a waste stream. Of course, further down the track, we might be able to harvest some benefit from that because, obviously from a sustainability standpoint, we'd like to be able to use those eggs, whether it's in industrial purposes or, they are fertile eggs that could go into things like vaccine production or possibly, protein harvesting or something like that. So there are possibilities, but, right now when we talk about how this could be implemented and certainly how it could be regulated, the first port of call is that will be a waste stream. That's our genetically, that's our genetically engineered material there, and that will go into waste. Or our females, which do not contain any markers and are not actually genetically engineered at all, they'll go on, they'll be incubated, they'll hatch, and then they will go on to lay eggs that people eat as table eggs. Okay, so how does this actually work? And so in humans and mammalians as well, we have sex chromosomes, we have X and Y, and in chickens, they have Z and W. And so I guess probably a lot of you would be familiar with the idea of sex semen. So in things like the dairy industry, you can buy sex semen because males carry X and Y. And so that means in cattle semen or mammalians, males carry X and Y. And so in cattle semen, you have some semen contains the X, some semen contains the Y. You can basically bind some stuff to it and differentiate your X from your Y. Chickens are the opposite of this. And so here we have our rooster and he carries Z and Z. So you can collect semen from him, but all of his semen has Z's in it. So there's no differentiation between that. And so the sex determining member of the species is actually the female. So she has a Z and she has a W. So she's actually in charge of determining, what the sex of each of those eggs are. And so what that means is that if we have an animal where we have a male that has 2 unmarked Z chromosomes, and we breed him to a female that has a marked Z and an unmarked W, this is what their progeny will look like. So the male is always contributing a Z, so he's contributing unmarked Zs. The female, if she's going to have a male offspring, contributes her marked Z, and so that comes over here, and so that allows us to sort those males out, whereas if she is going to have a female offspring, she's going to contribute her unmarked W. And so basically what we're doing is we're relying on the idea of, segregation of the sex chromosomes to produce this null segregant event where we have marked males, they're genetically engineered, we pull them out of the system, and we have unmarked females, they're not genetically engineered, they don't contain any genetically engineered material, but they're actually the ones that go further along the system and actually, contribute to the food products because they're the ones that are going to lay the eggs. And so it's using genetic engineering within the breeding program, but it's not actually using genetic engineering at all in the final food product. Okay, so what does this look like at sort of a larger scale? And so Hassa very nicely had a pyramid up there before, so I won't go into super detail on this, but basically the way that the poultry industry works and a lot of other industries work is that you have a crossbreeding system. And so we have 4 lines here, A, B, C, and D. And so to integrate this into the system, we start with the C line. And so up here is our top pedigree animals, our very fancy and special animals. And so in that C line, you don't have to touch A, B, or D. You just have to think about C. And so we have, to do this, you need to have double marked males and single marked females. So then you crossbreed those to get your double marked male here at the grandparent line. That will eventually allow you to produce your single marked female, which is the one that we were looking at before, which when you cross with an unmarked male allows you to make marked male embryos and unmarked female embryos. And so that's how this sits within the breeding system. And then just sort of zooming that out from the crossbreeding out to the pyramid. And so as Hassa explained before, at the crossbreeding, you have 4 different lines that eventually you're breeding into one line, but it's sort of the reverse. As you go down the pyramid, you start with a very few animals and then you breed out to lots and lots and lots of animals. And so we have our pedigree selection and our genetic improvement here. And this is where you actually integrate the marker. So you fix that trait up into your pure line stock. So you're not constantly having to reintroduce it. You fix it into the stock. And this is where your pedigree selection is happening. And then once it becomes a fixed trait, then you just breed it down. So into your grandparents and your parent stock. And then here you can see this is where our null segregation event actually happens. And so this is where your marker exists within your breeding pyramid. And then you have your null segregation event. And then everything going down from there is just your commercial layers, which, as we talked about, don't actually contain the marker. And then your commercial layers are producing your eggs, that are going to processors and retailers and consumers. And so you have the technology sitting at the top of the pyramid, but when it actually gets down to the commercial production, that technology is no longer in the breeding system. So how do we do this? Simon actually already talked about this as well, so I don't have to go into nitty-gritty details, but as always, it's a science talk, so I feel like I have to throw a little bit of technical science in there. So making genome engineered chickens is a little bit different than making it in mammals. As Simon explained, you don't actually have access to the single cell zygote, and so we have to access the germ cells through primordial germ cells, and so we collect embryos. Our research partners at CSIRO use a process known as direct injection, and so we directly target the primordial germ cells as they are migrating to the gonad with our genome engineering tools. Those animals go on, seal up the eggs, and you hatch animals. And so from there, what you have is animals that have a mosaic gonad. And so some of the PGCs that eventually become the sperm you have targeted, some of them you haven't. You take those males and you evaluate them to see what percentage of the sperm in each of the males has the mark that you're looking for. And then you build a breeding program off of those animals. And so that's sort of the technical detail of how we do it. Obviously, there's a lot more complicated things in there as well, but that's the nuts and bolts of how it works. And so that's it. I'd like to really thank the team at CSIRO, without whom none of this happens, and take any questions.